That was a great introduction. We have the slides. Thank you, Dr. Kenny, for inviting me to present this talk, and I guess there's a way to move this. Wonderful. I'm going to talk a little bit about the biophysical mechanisms underlining riboflavin cross-linking, and then briefly show you some of the major hurdles to riboflavin cross-linking that was sort of mentioned by Dr. Farid, and then some applications using femtosecond lasers to use two-photon nonlinear optical approach to improving riboflavin cross-linking. This whole story begins with a question as to what controls corneal curvature, and I think this also goes to the question of what is the underlying functional problem in keratoconus as well as post-LASIK ectasia. The shape of the cornea obviously defines the refractive power of the eye, and its ability to maintain that shape determines the visual acuity back in the retina. The shape of the cornea is thought to be controlled by the biomechanics, predominantly involving the corneal stroma, which is 90% of the thickness, which is composed, but again, predominantly by fibrillar collagen, which is organized. These fibrils are organized into bundles, which we call lamellae, which are ribbons-like things, and the weaving of these ribbons then control the shape and the mechanics of the cornea. We've been using nonlinear optical imaging of second harmonic generated signals, which is shown here. These lines then correspond very nicely to the collagen fibrils that are seen by transmission electron microscopy. Of course, these are nanometers, these are submicron images, but you can see that fibers running in plane show up very nicely, and then we can obviously take multiple images over the entire cornea and generate these three-dimensional Google maps of the corneal stroma, and then follow individual fibers as they branch in anastomose. So there's a branching, fibrils are going now in two different directions. And using this type of imaging, we've tried to reconstruct the cornea throughout its various layers, particularly, and one of the things we've found is that the cornea is comprised of a complex array of branching and anastomosing of these lamellae, particularly in the anterior part of the cornea, and then this decreases as we go to the mid and the posterior cornea. And we've quantified this when working with the computer science group to automate this. But again, it's very high in the anterior, drops in the posterior. And if we cut the cornea into thirds using flaps, interlaced flaps, and then do mechanical testing, we find that the stiffness of the anterior cornea coincides with the amount of branching that occurs. To further emphasize the importance of branching in the structure of the cornea, we've looked evolutionarily at the development of this lamellar branching, looking from sharks, bony fishes, amphibians, alligators, birds, and mammals of all different sorts. And one of the things we found is that when you look at the fish, it's a very simple cornea. These collagen fibers are actually organized into a sheet that goes from limbus to limbus straight across the cornea. And each sheet is then rotated, and it looks very much like plywood, which it is, essentially a protective cover. But as you walk up the evolutionary tree in these extant species, you begin to see these branching of fibrils extending from one lamellae to the next. This becomes more prominent as you go to amphibians, reptiles, and most dramatically by birds. And that's shown here in this reconstruction, which this organization takes on a chicken wire type appearance, in which then the cornea is incredibly stiff. This also coincides if you do the quantitation. If you look at fish, there's low branching and it goes up from bullfrogs and very high in birds. And of course, birds have the highest visual acuity of any vertebrates, including humans. If we move on then to look at this paradigm in the sense of keratoconus, and I think that's been wonderfully introduced by Dr. Farid, but we've done a second harmonic imaging of the corneal, of the cone and keratoconus, as well as the periphery of the keratoconus. And what we find is a simplification in the lamellar complexity. No longer do you have the kind of branching that you see in the normal cornea in this cone region, yet you do pick up branching in the paracentral region. And this is quantified here looking at branching in normal versus keratoconus. And in the anterior part of the cornea of keratoconus, there isn't even as much branching as you see in the posterior normal cornea. Interestingly, also in the central cornea, normal, it's the same amount of branching centrally as peripherally, yet in keratoconus, it's much less in the center cone region than out in the periphery. Mechanically, using acoustic radiation force elastic microscopy that we've done, we've seen similar huge drops in the elastic modulus in the center and in the anterior and posterior part of the cornea, and this coincides with Boolean microscopy, looking at the mechanical stiffness three-dimensionally in the cornea. So nature, in order to control shape, has woven collagen into certain shapes that allow for stiffening of the tissue. It's not done exactly by cross-linking. However, as mentioned by Dr. Farid, Wollensak and Sporel some 20 years ago suggested that we could do this chemically by using riboflavin, and she's already excellently described this. Essentially, when you excite riboflavin, it goes to an excited state. It can also fluoresce, but it can also transition to an excited triplet state in which it can donate an electron, form oxygen-free radicals, and then induce cross-linking from tissues. This cross-linking can be seen using blue excitation light as blue collagen autofluorescence. And normal tissues during aging achieve this blue collagen autofluorescence. In these regions of blue collagen autofluorescence, you can measure a stiffening of the tissue, and indeed also another thing you can look at, there's a decrease in the crimping. Normal collagen in the cornea, these are SHG images, has a sort of waviness pattern to it, and you can measure how much waviness there is by measuring crimp. And in regions where the cornea was cross-linked, it's now straight. And we believe this has something to do with shortening of the fibrils, stiffening of them, and leading to flatness of the cornea. So what are the problems? Again, Dr. Farid mentioned these, but I'd like to, besides delayed, I'd like to mention that UVA cross-linking results in a marked killing of everything in the cornea, particularly in the rabbit. The anterior stroma, this is 1 month after surgery. The blue here is the collagen autofluorescence. The green are the live cells that are present. And you'll see even in the posterior cornea in the rabbit, these cells are gone. Even at 3 months, there's no cells in the rabbit in the anterior part of the cornea where there was cross-linking. And of course, this type of delayed wound healing leads to other complications. In addition, the cells migrating in that you see here induce a lot of light scatter, looking at this cross-section of light scattering from the cornea that can be measured. And then also, it's very difficult to actually pattern your cross-linking using UVA light. So therefore, we've tried to look at two-photon excitation, nonlinear optical excitation of riboflavin. I think everything is sort of told in this particular picture. Here's single photon shining a UVA light into a vat of riboflavin, and you can see that the light is blocked the minute it gets into the riboflavin. So in a cornea, only the anterior part of the cornea is actually being cross-linked, and the depth of penetration is very limited. With two-photon excitation, you focus the light, and only where the light becomes in focus, with infrared light, do you get excitation, and you can see the fluorescence. And that would be the region where you get free radical generation and cross-linking. So what we've done is build a nonlinear optical cross-linking delivery device. This includes a beam expander and an objective, and allows us to vary the numerical aperture of the objective. This objective focuses light down on an applanating cone, and we have a rabbit eye here. We've also put rabbits under this to do cross-linking. Changing the numerical aperture of the objective, we change the focal volume. These are the axial fluorescent images in a riboflavin taken with a camera. You can see this is about 150, maybe 200 microns long and 2 to 3 microns wide. By changing it, we can change the length of that within the tissue. We can also vary the depth, so we can create any kind of geometric pattern we want inside the cornea. So in order to generate the light that we need 760 nanometer light, and we've gone to amplified pulses so we can use microjoules rather than the nano picojoules that are available with most standard femtosecond lasers. But to get this amplified laser light, we've had to build a non-collinear optical parametric amplifier, which is this, and I won't get into the guts of this, but basically we generate white light using this 1030 infrared light by passage through a sapphire plate. This white light then has all the spectrums of femtosecond laser light, and you can see this white light here as it goes through this dispersion chamber of water cuvette. And then we select out the specific frequency at this BBO crystal that we're interested in, which is the 760. This is looking down on the surface of the cornea, and each of these spots represents a region of collagen autofluorescence that was achieved by one single pulse of this femtosecond laser light done at 5 kilohertz per second. And then these are 3 passes through the cornea cut and cross section. We've also done this in rabbit eyes now. We did a series of about 11 eyes, 10 rabbits. This is the eye 2 days after. This is 2 weeks after we took the eye out, did a section. You can see here the collagen autofluorescence. It spans about 110 microns deep. It's down 90 microns from the surface of the epithelium. And this is the change in corneal curvature over time in these rabbits. There's a marked drop at 1 week, then it levels out by 2 weeks, and we think this has to do with having to remove the epithelium. But we get about 1 to 1.5 diopters of flattening consistent with what you see clinically. We're also moving on trying to improve the epithelial penetration of riboflavin. We have an enhanced riboflavin penetration solution. This is the Dresden protocol, and comparing the amount of riboflavin that gets into the stroma with about EP2. It's about equivalent to that which you can get in the Dresden technique. This is after this EP2 exposure. You see no damage to the corneal epithelium in this cross-section, no damage to the stroma in this live dead. These live cells are green, also the corneal endothelium. We can also cross-link. This is the region of cross-linking. This was 24 hours after nonlinear optical cross-linking. This 100 microns are where we cross-link. The overlying epithelium's normal, and the underlying endothelium and the keratocytes are all normal. So we think this has great opportunity for the future. I'd like to thank then my contributors, Dr. Brown and Dr. Uhas, who's done a lot of the femtosecond laser work. And I'd also like to recognize Sam Bradford, who's a graduate student in my lab in biomedical engineering. Eric McCula worked on the NOPA. Dr. Sung, who did the refractive analysis of the rabbits with the orb scan. And Yilu Zai, who keeps us all straight and narrow and helps us find where things are in the lab. And thank you very much. I think people have done that. They've advocated using cross-linking for bullous keratopathy. It has a change in the water profile, so you don't get that edema and swelling underneath the epithelium. But I haven't followed that very much, but I think that's already been advocated and looked at, yeah. You know, it takes months for the wound healing process to, at least in the animals, and I think also in the clinical studies using in vivo confocal, there is also noticed a rather large delay in the repopulation of keratocytes in the anterior stroma that requires months, if not a half a year or more, to repopulate to a year. So you're dealing with an 8 millimeter diameter cross-linking region because that's the area that you remove. If your button's larger, you wouldn't have that problem. If your button's smaller, you may have some problem in not having the keratocytes be able to move in to heal the wound as well. I would add the lens as well. Remember that the fish lens is very hard and the mammalian lens is very soft, and that the evolutionary changes that occurred in the cornea to allow stability and refraction, allowed the lens to change because now it could be used for accommodation, which then allowed the retina to become more of a sensor detector, a higher detector than it was in the fish. So the whole thing, I would think, in my view of it, evolved together. You can't have a high visual sensor detector in the retina if you don't have an adequate refractive system going from the cornea to the lens. So without the evolution in the anterior part of the eye, you don't have any evolution in the back. Well, if you can go to the birds have very interesting way. They can actually, with contraction of the muscles, pull down on the cornea and change the shape by 4 or 5 diopters, as well as the lens can push forward through the iris and have another 7 to 11 diopters for those kinds of birds that can dive into the water. So there's quite a bit of variation, evolutionary adaptation in both the shape of the cornea and how the cornea can change shape, and the lens as well.
It's the extraocular muscles that pull that can change because after
there's muscles that connect to the scleral ossicles, and that those ossicles then pull on the corneal stroma, which goes out to the edge. As you can see, there's the fibers go from limbus to limbus all the way across 360 degrees. So as those muscles pull, it pulls down and steepens the central cornea so that they can see better or at different times.
Stabilizing things that we did as a community to eyes was RK, which we don't do anymore, but RK continues to have destabilization and biomechanical instability years out with fluctuations, as you mentioned. Application for cross-linking in RK hasn't been studied too much, but I think that's a potential area in RK eyes. We hesitate to touch them with anything because we can further destabilize them. But in an eye that's stable, doing like a cross-linking procedure to stabilize that cornea. Natalie, do you know anything further on that? I don't think much has been done to look at that.
Well, I think that, the myopia research suggests that the sclera remodels depending upon its input from the retina. And so it continues to change over time and gets larger. So for progressive myopia, whether similar kinds of changes occur in the cornea, they don't have to require as much change to change the dioptric power. You only need 5 microns of flattening or 10 microns at most to get a diopter or more of change in the curvature. So, but generally it's not cross-linking, but in the sclera, it's a whole complete remodeling that's going on back there. And then, of course, people are now interested in using cross-linking to stabilize that, and they are doing that. There are complications associated with that to the retina, but that is being looked at. It's also being looked at for glaucoma, some way to get back there and alter the mechanics of the posterior pole so that you don't have the changes in the outflow canal of the optic nerve.
Thank you very much.